Hi, today we're looking at biomacromolecules. What you may notice is the picture on the front is actually a picture of a protein. It's made up of many or numerous proteins. You can tell that by the different colours that they've used to represent each of them. So that must make it quaternary structure. What you may notice is you've got these alpha helixes spread throughout and also some random coils. When you come across pictures like this, it's important that you try and break down what information you can gather from it, because it's an important study technique that will hopefully help you in your exam. So when you come across a picture, have a look at it. What information can I get from that picture? Then how can I use that to answer a question? So before you can actually look at what different types of biomacromolecules there are, it's important to know what actually is a biomacromolecule. In biology, you'll come across many big words. The trick is to break them down into their smaller components, and you'll find that you have a much greater vocabulary than you think you do. So biological, macro, the opposite of micro. We use a microscope to look at small things, so macro is looking at big things. A molecules is just a cluster of atoms. So it's a big biological cluster of atoms, a biomacromolecule. What they are is large molecules found in living organisms. They're vital in cellular structure and function, so how we are built and how we work. They are all organic compounds. When you see the word organic, we hope that you make the link in your head. If something's organic, it must have carbon joined to hydrogen. It usually also contains oxygen. They are all polymers, so they contain monomers, mono, one, poly, many, so monomers joined together. What is the exception? Lipids. They must be manufactured by cells, so they're made within the cell. Going back to protein synthesis, they form covalent bonds, covalent, sharing electrons, via condensation reactions. A condensation reaction, condensation is rain, so when the monomers join together, they release a water particle. The four classes of biomacromolecules that we look at this year are lipids, polysaccharides, nucleic acids, and proteins. Okay, so we'll start with the exception to the rule, the one that is not a polymer, a lipid. Before we look at anything else, it's important to think, what do I already know about lipids? So what did I learn in year 11 that could help me now? You would have learnt that lipids are hydrophobic, so they hate water. When you think about it, if you ever do the washing up at home, so you've cooked meat in a pan, you've got to clean the pan afterwards, you put the pan in the sink, you add water, you add detergent. Automatically, you can see the fat rises to the top. It is trying to minimize its exposure to water. So lipids are hydrophobic. They hate water. Their subunits, glycerol and fatty acids. So you see the word glycerol, you think it might be something to do with the glucose. So it's a glucose, or a form of glucose, or carbohydrate, with 3 fatty acid tails in this picture. Because this one has 3 fatty acid tails, that makes it a triglyceride, tri being 3. The phospholipid bilayer has 2 fatty acid tails. The chemical composition, it's organic, so it contains carbon and hydrogen. Usually they also contain oxygen. Lipids are no exception. Their biological significance, so what we need to know about them. They are an energy source and storage, so we use them for energy. When you see an energy source, do our bodies use fats directly to power our cells? No. We go through a process of converting ADP to ATP. So even though it's an energy source, your body has to go through a series of chemical reactions to access that energy. It's a major structural component of the membrane, so it keeps our membrane slightly fluid, and it functions in cell-to-cell communication and recognition. When we move on to the immune system towards the start of term 2, that will become really important. So just keep that in mind and store that in the memory bank for later on. There are different types of lipids. I mean, the picture before, you had a triglyceride, whereas in your phospholipid bilayer, you've got 2 fatty acid tails. So some of the different classes of lipids that we come across this year. So your fatty acids, their function or their purpose, use is energy source, and they're a subunit of other lipids. So they're a building block. Your triglycerides, fats and oils, They are energy storage. So fatty acids, we use the energy. Triglycerides, we store the energy. Phospholipids, that's a structural component of the plasma membrane. Just something to keep in mind, if you have to draw a picture of a phospholipid bilayer, each phosphate head must have 2 lipid tails to get the marks in your exam. Your glycolipids, again, you've got that glyco prefix, so it's something to do with a carbohydrate. They are recognition sites on the plasma membrane, so they help to determine self and non-self. So when we do the immune system later on this year, that will come into play a bit more. Steroids, including cholesterol and your sex hormones, only found in eukaryotes. They are a component of the plasma membranes and important signaling molecules. And finally, your terpenes, probably better known as vitamin A, they are an antioxidant. So here we have a couple of different pictures of lipids. So the top one up here, we've got a saturated and an unsaturated. The difference between a saturated fat, which you tend to find in animal meat, and an unsaturated fat in things like vegetable oil is one sets hard and the other one does not set hard. The reason why a saturated fat will set is because you've got this straight line here, so they can actually build in nice straight lines. Because unsaturated fats have this double bond here or a kink, they've got that fluid structure to them, so they can't actually set hard. So that is why vegetable oils are fluid, animal fats will set solid. Over here, we've got glycerol, which it is an alcohol. What you may notice, it's joined to a fatty acid chain. I've got an OH and an HO, so a hydroxyl group. I can look at this and go, I've got 2 hydrogens and 2 oxygens. If I take out 2 of those hydrogens and 1 of those oxygens, I get water, H2O. So it is a condensation reaction because a water molecule is released. If I want to break the bond, if it takes a water molecule released to join them, I could add a water molecule by hydrolysis to then break them apart again. So glycerol has 3 hydroxyl groups or OH groups. Therefore, it can only accept 3 fatty acids. That means there's no repetitive linkage, therefore it's not a polymer. So just a little bit more on your triglycerides. You may remember from a couple of slides ago, triglycerides are important in energy storage. So they've got a carbohydrate or a glycerol, on the end, just here, and 3 fatty acids joined to them. 3 fatty acids makes it tri. So fats and oils are important energy storing molecules. They are stored in adipose tissue. Adipose tissue is just connective tissue that stores fat quite easily. Adipose tissue is usually found sort of just under your skin, so your subcutaneous fat, or around your organs, a little bit in the bone marrow and some also in the breasts. It's important, it does cushion, but too much can lead to things like obesity. Unfortunately, for things like adipose tissue, a lot of it is genetically determined how much your body will store. Okay, so here we're looking at the phospholipids. These are the guys that you're probably most familiar with. From last year, we would have done a fair bit on the plasma membrane when we did diffusion, osmosis, so that's sort of how these guys tie in together. So what we've got over here, we've got our phosphate head. It's just phosphate with some oxygens joined. So there's 4 oxygens, 1 phosphate. The 3 negative, that just is the charge that it has. Don't worry too much about that. It's not chemistry. And I've got 2 fatty acid tails. So remember, in an exam, you have to draw phosphate head, 2 tails to get the marks. The one thing about the phospholipids, you've got your phosphate heads, they love water. Your lipid tails, they hate water. So they have a perfect arrangement. I've got my phosphate heads that live on the outside and my lipid tails that live on the inside. When you see pictures like that, think, okay, so where is my extracellular fluid? Where is my intracellular fluid? So eventually those guys would have to make a circle to surround the actual cell. On the outside of that cell is my extracellular fluid. Think E.T. E.T. lived in outer space. And my intracellular fluid, so we've got intra-school sports where we play against each other within the school on the inside of the cell. So now we're moving on to our polysaccharides. Poly, many, a saccharide is a sugar. Their subunits or their monomers are the monosaccharides. So mono one. If someone's got a monobrow, they've got one eyebrow. Chemical composition. They're organic, so they must contain carbon and hydrogen linked together. Organic molecules usually contain oxygen as well. Polysaccharides are no exception. So their biological significance, they are a major energy source and store. You need to know in plants it is starch, in animals it is glycogen. That comes up on exam questions quite regularly, often in the multiple choice. Their structural significance, so their cellulose in a plant cell wall, they are found in membranes combined with proteins, so your glycoproteins. Glyco, that glucose sort of subunit, proteins, chain of amino acids, and lipids, so glyco for your sort of glucose type of thing, and your lipids. Just to clarify, glyco is not glucose, but it's a way that you can link a familiar word back to something that you do know. Okay, so the carbohydrates. You've got your simple carbohydrates, better known as your sugar, so your monosaccharides. Glucose is the main source of energy for plants and animals. It is soluble in water, but it's too big to cross the plasma membrane. That's when you need to think, if it's too big to cross the plasma membrane, how would it get through? Because we know our cells need energy. Therefore, it has to go through protein channels. You've got this picture over here that if you're like me and you're not really that into chemistry, you think, oh my God, I've got no idea what that means. You will never have to draw a picture like that in an exam. It does help if you can recognize it. If you took out the letters on the outside and you just draw the hexagon, you'd actually know, oh, okay, that's a simple sugar. Look in A1, above where the whiteboard is, you've got pictures of those that have been looking at you for the past 12 months. Okay, so don't stress if you see pictures like these. You'll never have to draw them. You may have to recognize them. So look at, with your biomacromolecules, what are the differences in the pictures, and just store those somewhere in the memory bank. Your disaccharides. So di is 2. They are water soluble. They must be broken down for transport across the membrane. Okay, so moving on to your complex carbohydrates. They are polysaccharides. So poly, many, saccharide, sugar. They contain more than 10 monomers. The most common ones that you need to know this year are starch, cellulose, and glycogen. They are insoluble in water. When you think about that logically, your cells are bathed in extracellular fluid that has a high water content. Cellulose is the main component of a cell wall. If cellulose was soluble in water, your cell walls would constantly break down. So that's where sometimes you can use your logic to think, how, if I come across this in an exam question, I don't remember, how could I get myself back to a logical answer? Okay, so rely on your common sense. It will pick you up marks in the exam. It can't pass through plasma membranes. The OH groups or hydroxyl groups are adjacent monomers. They can react, so they eliminate a water molecule. What type of reaction is that? Condensation reaction, water molecules being released. If I wanted to join them up again, I'd add a water molecule by the process of hydrolysis, and they link the glucose monomers together through a glycosidic bond. So glycogen, that is energy storage in animals. So we store glucose monomers. It's stored in the liver and in the muscles. Once the storage has reached capacity, the glycogen is then converted to fat stores. So you've got so much space in your liver and your muscles as your storage for your immediate energy source, so your glycogen. Once you've filled those up, then you put it back into the major storage of fat. So when you eat something, your blood glucose levels rise, your body releases insulin. The insulin then converts that glucose into glycogen that then gets stored until you need it. Take note of the picture of the glycogen, what you may notice. So I've got these hexagons again. So every time you see a hexagon, think glucose. And finally, we've got starch. So excess sucrose or glucose in plants is stored as starch. So when we talk about starchy vegetables such as potatoes, that just means that they have a lot of stored glucose. Again, you've got these pictures of these hexagons. So that's telling you straight away, I'm looking at a carbohydrate. Sorry, one more. We've got cellulose. So it's a structural polysaccharide in plants. So remember, poly, many, saccharide, sugar. I will keep repeating the poly and the saccharide until you are sick of it, but the idea is it then sticks in your head. They are also glucose subunits. So again, I've got these hexagons. I see hexagons, I see a carbohydrate. They form fibers. In humans, they are indigestible, so they provide a source of dietary fiber. When you look at them over here, so you've got these really nice straight lines, which makes them very strong and rigid. When you think about going back to the fats, when you had your unsaturated fats, you had that little kink at the end. The kink's what made them fluid. So cellulose is the same sort of linear structure as a saturated fat. So they set hard. When you think about it, cell walls are strong. Okay, so here we've got the nucleic acids. So these are what form our genetic blueprint, or our DNA. Their subunits, or their monomers, are the nucleotides. I've got a picture here, which this was in something very similar in last year's exam, so the 2013 exam. Here I've got, my base. Here I've got a 5-carbon sugar. So it's got 5 corners that are where the carbons are found. And lastly, I've got a phosphate. So they run antiparallel. So the base always joins to the sugar. And then I've got a free phosphate on one end up the top, and the free phosphate on the other end is down the bottom. Where the free phosphate is, that is always the 5' end. So the chemical composition of a nucleic acid, they are organic, so they've got carbon and hydrogen. They've also got oxygen, which most organic molecules do. In addition to that, they've got nitrogen and phosphate. These, as well as sulfur, are probably the only real chemical symbols that you will need to become familiar with. The biological significance, so they're the genetic material that determines the inherited features of an individual. So basically, what you inherit from your parents is the DNA. That DNA is a blueprint for what you are to become. It's also really important because it directs the unique sequence of amino acids in protein synthesis. What's the most common type of protein that you guys have come across? An enzyme. Okay, just going back to the 5' and the 3' end. So as I said before, so at the end of each nucleic acid or strand, the free end is the phosphate. That is always the 5' end. It runs down to the 3' end. The one thing to notice is that they always run anti-parallel. So one is going from 5 to 3, the other is going from 3 to 5. When we look at DNA replication, start of term 3 from memory, that will become important then. So just to have a bit of exposure to it now, again, store it in the memory bank for later. Okay, so our information molecules, or our nucleic acids. The order of the amino acids in each unique protein is determined by the information stored in DNA and RNA. So what is the difference between DNA and RNA? DNA is double-stranded. DNA has a deoxyribose sugar. RNA just has a ribose sugar. DNA has the base thymine, RNA has the base uracil. So again, when you see DNA and RNA, study techniques, go through what do I already know, so that way you're constantly revising what you have learnt. DNA is found in chromosomes, so there's 2 chains and it's got the double helix. They run anti-parallel, so the 5' to the 3', the other side is the 3' to the 5'. How do I know which end's which? The free phosphate is where the 5' end is. The base is always joined to the sugar. There are 3 types of RNA. There is mRNA, or messenger RNA, that is produced as a template against DNA. You've got tRNA, or transfer RNA, so it's involved in producing amino acid chains during protein synthesis. So it transfers amino acids from one place to another. And finally, we've got rRNA, or ribosomal RNA, which is a structural component of ribosomes. And here we've got the 2 different types of nucleic acids. So my deoxyribose and my ribose, so my DNA and my RNA. The subunits, so sugar, phosphate, and a nitrogen group or base. Either word is fine. So make yourself familiar with this picture. That way, in an exam, you'll be able to identify it. So phosphate, 5-carbon sugar, so it's got the 5 points, and your base. The nucleotides, they vary due to the structure of their nitrogen base. So in DNA, there are 4 bases. There's adenine, thymine, guanine, and cytosine. You must know how to spell them correctly. If you forget in the exam, go back through your multiple choice. Chances are they've already been written in there. Always remember, A pairs with T, G pairs with C. The way I used to remember it, my straight line letters go together, my curved line letters go together. So A and T have straight lines, G and C have the curve. And in RNA, I've got 4 bases again, but instead of thymine, I've replaced that with uracil. So A goes with U, G goes with C. It's important that you recognize when you get a sequence, whether it is DNA or RNA, because often that will be used to read off a code from the amino acid chart. So make sure that you are sort of looking at that, because that is one place where kids get caught up in exams. Okay, so what we have here is the DNA polymer structure. So nucleotide monomers, they are linked by condensation reaction. So every time they join, a water molecule is released. A phosphodiester bond is formed, and the polynucleotide extends in the 5 to 3 direction. So remember, my 5 end has the free phosphate. You've got complementary base pairing between bases, and they are held together by hydrogen bonds. So going back to sort of your biochemistry, your hydrogen bonds aren't that strong. That's important with DNA because I need to be able to make copies of it down the track. So when I make a new cell, I have to copy the DNA. That happens during mitosis and meiosis. Meiosis in the sex cells, mitosis in all the somatic cells or the body cells. So I need to be able to break them apart quite readily when I need to. One thing that you may notice, I've got half of my bases, so my T's and my C's, they have one circle, and my other bases, my A's and my G's, they have the 2 sections. If they've got 1, they are a pyrimidine. If they've got 2, they are a purine. I've always got a purine or a pyrimidine joining up together. The other thing that you may notice in this picture down here, I've got my G and my C have 3 sections where they join, and my A and my T have 2 sections. The way that I still remember it to this day is GC3. So my G and my C have 3 bonds, my A and my T have 2. Okay, so the proteins. To be honest, these are probably my favourite biomacromolecule. Don't know why, I just really like learning about these guys. So their subunits or their monomers are amino acids. Their chemical composition, they are organic, so they've got carbon and hydrogen, also usually contain oxygen. These guys also have nitrogen, phosphorus, and sulfur. Their biological significance, these guys are really important. So they catalyze reactions, so they speed up reactions by lowering the activation energy. They're signaling molecules, so hormones and receptors. They can be structural, so cytoskeleton and fibrous tissues, and transport, so things like protein channels, hemoglobin for oxygen transport. Every time you see a picture that has a protein in it, think, what level of structure is it? For this guy, I've got a chain that looks like a chain of beads. Therefore, it must be primary protein structure. So they've just been joined together. So with our amino acids, there are 20 different types. So I always think of it like 20 different letters in an alphabet. They are linked to amino acids, form a polypeptide chain, so a chain of many peptides. Proteins differ in the number and sequence of amino acids. So when you think about the English language, our words differ in the number and the sequence of the letters. So proteins are just a language using amino acids as their letters. The order of the amino acids is determined by the information in the cell's DNA. So your DNA tells your body what proteins it needs to make. If the DNA gives a wrong instruction, that means that your protein may not be produced or may not work properly. Some essential amino acids are not produced by the body and they must be taken in as food by animals. So when you hear in your popular media talking about essential amino acids, they're things that we must obtain from the diet to function efficiently. Just looking at this picture down the bottom, so you've got a set of amino acids that just look like a chain of beads, so it's primary structure. The double S here, that's just telling it's a sulfide bridge. Again, you'd be very unlucky to get that on an exam. You'll probably be able to recognise it by the end of this year, but it's one of those things that unless you're going for an A+ and a high A+, you're not going to be asked to draw one. With proteins, it's important to differentiate between biological language and language in the common media. So in the common media, when we talk about things like protein powders and protein from meat, in biology, we're looking at a sequence of amino acids. So it is important that you separate your biological definition from what you read on the internet or in newspapers, magazines. They're totally different meanings. Okay, so this is a picture that we generally use to show you that something is an amino acid because they have the same basic formula. You've always got a carbon in the middle and a hydrogen down the bottom. You've got a carboxyl group, so COOH, and an amine group, an NH2. So that's where your nitrogen comes into play. Your R group in an amino acid is what gives each amino acid its unique characteristics. So whether it's hydrophilic or hydrophobic, whether it's an acid or a base. So if you see a picture like this, the clue to you is it's got an R group, therefore it's showing me a picture of an amino acid. Make sure that you can recognize this. Okay, so you've probably noticed with all our biomacromolecules, they're all joining through condensation reactions. So in this picture here, so down the bottom, I know it's an amino acid. I've got an R group, so automatically I think, okay, that's a protein. So I've got an OH group here and an H on the end here. I think I've got two H's, one O. What's going to happen? Okay, so we know I've got two H's and two O's, so automatically I think, okay, that's a water molecule, so chances are those guys are going to join together. And here we see the water molecule has been released. And now I've got a nice peptide bond. So a water molecule was released and where I had the carbon and the nitrogen, those guys have joined together. You don't need to know it's a carbon and a nitrogen joining. All you need to know is it's a peptide bond. And again, just to sort of show you that it's an amino acid, I've got my R group, I've got my amino group, and I've got my carboxyl group. You don't need to know the chemical composition of the amino group or the carboxyl group. You just need to be able to recognize where they are in relation to the R group. So amino group, left-hand side, carboxyl group, right-hand side, R group up the top. Okay, so the importance of protein structure. So each protein molecule has a characteristic 3D shape, three-dimensional, that results from coiling and folding of the polypeptide chain. The function of a protein, or its job, depends on the actual shape of the molecule. And it makes sense that the actual shape of something will fit its job. Our bodies are quite efficient. We've sort of covered that in year 11 as we've gone through different topics. Proteins no exception. There are 4 levels of structure. They follow the exact same sequence as your schooling. You go to primary school first, you're currently in secondary school, a lot of you will head off to tertiary education, and then think of quaternary as like doing your PhD. This is where you can get really simple marks on exams. You often get asked a question about the levels of structure. Protein folding gives rise to the unique shape of a protein, and it is the shape that determines its functionality or its job. Okay, so first of all, we've got our primary structure. So we've just got our chain of amino acids. It's just like, a beaded necklace. Secondary, so that is determined by the characteristics of the R group. So there on the top, I've got my amino group on the left-hand side, my carboxyl group on the right-hand side. Hydrogen bonds form between close segments. We already know from hydrogen bonds, they're strong, but they can be broken. I've got my alpha helix chain and my beta pleated sheet. Also, I've got random coils. In an exam, you must write alpha chain, you must write beta pleated sheet. It is important that you get the alpha and the beta in there. It's not enough to write helix or pleated. You do need to write in the additional information. And just going back to the primary structure, so every time you see a chain of amino acids, think, okay, what type of bond do I have here? I have got a peptide bond. How is that peptide bond formed? By condensation reactions. So a water molecule is released that joins them together. If I want to break the bonds, I must add a water molecule back in. That is the process of hydrolysis. And here is just a different style picture. So I've got my alpha helix, my beta pleated sheet, and my random coil. In an exam, you can use the symbols for alpha or beta, or you can write the whole word. Pick whichever one is easiest for you. What I would do, choose one and run with it. So over the course of the year, either use the symbol or write the word. If you're consistent in the exam, you're not going to second guess yourself. Okay, so with our protein structure, we're looking at tertiary. So I've already got my chain of amino acids. That's my primary structure. Then I've got my alpha coils, my beta pleated sheets. And Then they fold into a three-dimensional group. So it brings the R groups close together so stronger interactions can occur. These interactions determine the functional shape of what the protein actually looks like. What you may notice in this picture, you've sort of got a couple of different types of bonds. So I've got a disulfide bridge. All that means I've got a couple of sulfurs linked together. I've got hydrogen bonds. The one thing to know about protein structure and why they fold in certain ways, if I've got one of my parts of the chain that is hydrophobic or it doesn't like water, that's going to fold itself towards the center. My hydrophilic parts, so the parts that love water, they're going to be on the outside because our cells are quite an aqueous environment. There is a polypeptide backbone, so polypeptide, a chain of many amino acids. So these guys keep folding based on their DNA instructions, and at this point they're called globular proteins. So to me that's like big blobs, so globular, and they usually make enzymes. And finally we've got our quaternary structure. So this is where we've got proteins that are in the tertiary structure that join together to make a monster polypeptide chain. So if you get asked for an example in the exam, hemoglobin is probably the most easy one to remember because you've come across hemoglobin in the past because it's part of your red blood cells. So the same sequence will always fold the same way. So it's like in our language, we've got ways of spelling words. DNA is the same. So if the DNA instructions say the same thing, the protein will do exactly the same because it's got the same set of instructions. So what you can see here, I've got 4 different amino acids that have joined together. And with hemoglobin, not sure if you know this, but they have the carrying capacity for 4 oxygen molecules. So one thing about proteins is they're often produced in an inactive form. So our body doesn't have them transporting around in active form because then they might come into effect before we need them. So usually they require an enzyme to convert them to active form. So we've got proteins in our body that require another protein to almost flick the switch to say, I need you to work now. An example that probably makes sense is insulin. You don't have insulin constantly flowing through your body or else your blood glucose levels would drop too low. If you alter one amino acid in a chain, then the folds will alter. One example is thalassemia. That's, one amino acid that's changed and that alters the oxygen carrying capacity of human red blood cells. When you think, what are the consequences of that? Think of cellular respiration. If I can't effectively carry oxygen to my body, then I can't convert energy as efficiently as I may need to. If something is conjugated, it is joined. So it's proteins bound to other molecules. So things like hemoglobin and nuclear proteins. So nucleo is ribosome, so it's protein and RNA joined together. The nucleo is like a nucleotide. Okay, so what is the proteome? This has come up in exam questions. So it's most chemical reactions involve proteins. That you know because enzymes are required to lower the activation energy so our bodies can work efficiently. The complete set of proteins produced by a single cell or an organism is the proteome. The same as the complete set of genes produced by a single cell or an organism is the genome. No protein acts alone. So these days, one area of biology in particular that a lot of research is going into is studying the interrelationships between proteins in a systematic way. So that is proteomics. So the study of the proteome. So if any of you are interested in sort of a biological career, that is one area that work is going into at the moment.